Assalamu alaikum and welcome back to another video. In today's video, we are going to discuss about another technique that is gel mobility shift assay, also called as the electrophoretic mobility shift assay or EMSA. Now, why this technique and what are the uses of this technique? Let's understand. Whenever we have a DNA and a protein interacting together and you want to see the interaction between the two, that whether our DNA and protein is interacting, in order to figure out that interaction, we use this technique called the electrophoretic mobility shift assay. From the name itself, we can deduce that if you have a DNA, just the DNA, and you are running it on the electrophoretic gel, what you will see is its mobility, it will be faster as compared to the DNA which is bound to a protein. And that protein, it can be anything. So because now it will be having higher molecular weight because of the bound protein. So as a result, it will take time as compared to that of the naked DNA to get to the bottom of the gel. So that is the principle of the electrophoretic shift or the electrophoretic mobility shift assay. So the detection of the DNA-protein interaction, it relies on the fact that a smaller DNA, it is going to have a much higher mobility in the gel electrophoresis than the same DNA goes when it is bound to the protein. It's a simple concept which is used in this technique. Now to make you understand, we'll take this gel here. In the lane 1, what we have is, we have just, so you can see here, this is the representation of the bare DNA. So in the lane 1, which we can also call as, it serves as a control. Now you can see the mobility of this. And now you have the same DNA. So the same DNA, but now this is linked to another protein. It's forming a complex. And now we have a DNA and a protein complex here. So this is the case here in the second and in the third. Now you can see in the first case, you had the DNA and it's running in this speed. It will be the first to reach the bottom of the gel. But when the same DNA it's complexed with a protein, it's forming a complex, it will take time. Its mobility in the gel is reduced. Now, see the third component here. So this is a DNA, double-stranded DNA, which is bound to two different proteins. Now that may be the two proteins directly interacting with the DNA, or it might be one of the proteins interacting with another protein. Now in that case, still, this complex, it will have higher molecular mass, and when you are running it on the gel, see here. So this is here. When this DNA bound to two different proteins, you will see that there will again be a shift in the electrophoretic mobility. So that's called as the super shift. So if we sum up this technique, it's just a technique to see the interaction between the DNA and protein. Now let's consider if you are looking for a protein. Now, where do we use this technique? So in that instance, suppose you have a DNA and you want to see whether or not the different transcription factors, they are binding to the promoter, they are binding to a particular region of the DNA. So you'll use this technique of electrophoretic mobility shift assay, which will give us basically an idea that whether, so if there is a shift, that means the interaction is happening between the DNA and the protein. So that is all about this technique of gel mobility shift assay.